Hello again. I am wanting to continue now with the study of cereal dilutions and the milk lab. So in the milk lab, what we would have done is had the dilution blanks, which contain 9 milliliters of sterile water. Into those, we would have put the initial 1 milliliter of our milk sample, whether it was the outdated or the fresh milk. That would have been the first 1 to 10 dilution. Then we would have taken 1 milliliter of that solution and put it into the next 9 milliliters of sterile water, and that would have been another 1 to 10 dilution, bringing us to a 1 to 100 dilution, etc. So we talked about the cereal dilution last time, and you know that we would have had a 1 to 10 dilution in the tube with 1 milliliter of the milk and 9 milliliters of water. So then the 1 milliliter was taken out of each of those tubes, put into the agar plate. Well, it would have been just a blank plate at first, and then the liquid agar was poured on top and allowed to settle and congeal, and then we incubated the plates. So what I've tried to simulate here is what the plates might have looked like, and I'm showing you what is a possible result from an outdated milk sample. What you're seeing here in the undiluted milk sample is an incredibly high number of colony forming units that would literally be impossible to count. And as I was mentioning in that last video, I told you that we use a term too numerous to count, or TNTC. So that is how that plate would be recorded, as TNTC. So the undiluted plate is TNTC. Notice that we get the similar result in the 1 to 10 dilution. You can see that there's been a decrease in the number of organisms, but still there are so many that it is too numerous to count. The same is true at the 1 to 100 dilution. Again, you can see that the dilution is working. We've definitely decreased the number of organisms in this third or 1 to 100 dilution. Well, it's actually the second. But the number of organisms that grew on the plate are still too numerous to count. Now look what happens in the 1 to 1,000 dilution. You can see that 39 CFUs are on this plate, whereas on the 1 to 10,000 dilution, there are 11. Now, let me briefly explain CFU. That stands for colony forming unit. We normally assume that an isolated colony grows from just one organism landing in that particular spot on the agar plate. And we know that that's not true. There are times when it could be diplococci or streptococci or diplobacilli, so that there is more than one that actually formed that colony. So to account for that, we use the term colony forming unit rather than saying that there are that many colonies, meaning that they all arose from one single bacterium. So on our 1 to 1,000 plate, we got 39 colonies. On the 1 to 10,000 plate, we got 11 colonies. Now, which is the plate that we go by to determine how many organisms were in that original milliliter of milk? Because that's the whole point of doing this, right? We wanted to know how many organisms were in that milk sample. So we took 1 milliliter out, we put it into the first dilution blank, making our first 1 to 10 dilution with 1 milliliter of milk. So our results then are going to be how many organisms were in that 1 milliliter of milk that we used to start this. A lot of people immediately think that we ought to use this plate because it has an easily countable number of organisms. But let me show you something. If I take a tube and I have very few organisms in it. And of course I'm exaggerating here. But if I take that tube, depending on where you put the tip of the pipette, you could distort how many organisms are in that tube. If you drew from this section, you would get barely any. If you drew from down here, you would get more, but not many. And if you drew from up here, you would get quite a bit more. So they've done statistical analysis on this and figured out that when there are fewer than 25 organisms on the plate, or some labs do use the level of 30, but when there are that few number of organisms or CFUs on the plate, it's inaccurate to count it. There are too few to use that plate. So for our purposes, we're going to stick with 25 to 250 as our range. So we can only use the plate in this serial dilution, or from this serial dilution, that has between 25 and 250 colonies. And of course, when you look at our results, the only one that we can use is the 1 to 1,000 dilution. That plate had 39 CFUs. So would we report that the 1 milliliter of milk had 39 CFUs per milliliter? Hopefully you're thinking, that doesn't sound right, and you'd be right. Because remember, we diluted the milk to get to that plate. What we have to do is take this number and multiply it by the dilution factor. So 39 times 1,000, or 39,000. So our result for this particular serial dilution would be that there are 39,000 CFU per milliliter. And that is what we would report from this particular serial dilution. So hopefully that helps you understand how we interpret the serial dilution, and with the two videos together, I hope you get a sense of what the milk lab would have been like. It's actually a very fun lab to do because you get to not only do the serial dilutions, but you get to do the pour plates. And I am sorry that you weren't able to do it, but I really hope that what you learned is the takeaway of why we do a serial dilution, how to do them, and then importantly, how we use the data that result from those dilutions in order to determine how many organisms were in the original milliliter of milk. Okay, thanks. See you online.